Vivo, doutora,
Yeah.
pode iniciar.
Ok. So, gracias y bienvenidos a todos a este nuestro segundo encuentro de IMMUNOLAC, que traz charlas e discussões de científicos imunologistas de Latinoamérica e Caribe. Muito obrigada a todos pela presença nesse nosso segundo encontro IMMUNOLAC, trazendo hoje pesquisadores da América Latina e Caribe mais abertos para todo mundo. So thank you everyone for the presentation. Now we are going to switch to the official language of IMMUNOLAC, which is English. And I would like to first thank the participation of Drs. Mariana Salatino and Ken Gollob. Mariana is from Argentina and Ken is from Brazil. And also Gisela Suárez, who is coming to share the coordination of the presentations with me from Cuba. So we are going to start with Mariana's presentation, and I will introduce her. And then after Mariana's talk, we'll have questions for her. So you guys feel free to place the questions in the chat in the YouTube. And after Mariana's talk, we'll pass it on to Ken, who will be introduced and coordinated by Gisela. So it's my great pleasure to introduce Dr. Mariana Salatino, who obtained her master's degree in science and a PhD in molecular biology at the University of Buenos Aires in Argentina, where she studied the crosstalk between progesterone receptors and breast cancer. In 2006, Mariana started her postdoc under the mentoring of Professor Rabinovich, which is well known by most of us here, studying the impact of galectin-1 on cancer progression with special attention to tumor immune escape. At present, Mariana is an independent researcher of CONICET at the Institute of Biology and Experimental Medicine. And during the last 10 years, she has led the breast cancer group in the Institute, focusing her attention on how galectin and glycan interactions control metastasis by shaping the immune response in breast tumor microenvironment. During her career, Mariana, who is very well accomplished, gained many awards and honors, such as, for example, the AACR International Scholar Award, the Conex Foundation Award, the LAUSEC Award, and Esteban Montuori Prize. In addition to her many important scientific contributions, Mariana also has been contributing quite significantly by mentoring several students and postdocs in Argentina. So, gracias, Mariana, por tu presencia en nuestro encuentro. And please take it over from here.
Gracias, Wal. Let's put the screen here. Can you see it? Yes.
Yes.
Well, thank you all. Thank you, Walt. Thank you, Ken. And thank you all for the invitation. For me, it's a really pleasure to be here in behalf of the Argentinian Society of Immunology. So I want to show my results, and we will discuss the opportunity to treat breast cancer with immunotherapy. That is not an easy task, because breast cancer was not the typical tumor that is treated with immunotherapy. So I will tell you why. Okay, here. So breast cancer is a very common disease, but the good point that is curable when it's detected early. Okay, and the problem is the metastatic stage. That is here, yes, the metastatic stage. So a normal mammary gland is like this. It has luminal cells, it has myoepithelial cells, and a basement membrane. And here, when there is a mutation and initiate a proliferation, it goes first to a typical hyperplasia. And then if this proliferation continues, it goes to an in situ tumor that is surrounded by the membrane. But if this proliferation continues, it invades the surrounding of the tumor and it goes to an invasive stage of the tumor. And if it keeps progressing and invading, it goes to a metastatic stage. And this is the problem, the metastatic stage in breast cancer, because it can go to secondary organs, and it can cause the death. Okay. Well, during progression, immunodeletion occurs. And immunodeletion, as you may know, is called the resulting tumor. Immunodeletion is composed of 3 phases: elimination, equilibrium, and escape. During the elimination phase, the immune system is completely successfully to eliminate the tumor cells. So antigen-presenting cell detects the tumor antigens, and CD8 and CD4 T cells can attack and proliferate and destroy the tumor cells. And also the macrophages can engulf the neoplastic cells. Then elimination can go to the equilibrium phase, that is an intermediate phase that is occult, and the final number of cells is always the same. It's like an unstable stage. And then in this, it can go then to the escape phase. That is when the tumor is detected. During the escape phase, the tumor is clinically detected, and many mechanisms of escape, of immune escape, has been described. And in this point, the immune response is not able to counteract the tumor progression. So, for example, you have infiltration with suppressive cells, you have a failure in the antigen machinery, and you have the expression of immune checkpoints, inhibitory immune checkpoints as CTLA-4 and PD-1 and PD-L1. So this is, we have here an immunosuppressive microenvironment in contraposition to the immunogenic microenvironment that you have when you can eliminate the tumor, okay? So if we make focus on the elimination phase, that is when the immune response is able to eliminate the tumor, you can see here that this is a melanoma, but we can imagine that this a breast tumor is the same. So tumor antigens are detected and are captured by dendritic cells in the breast. Dendritic cells then travel to the nearest lymph node where the specific lymphocytic activation is produced. Then the naive T cell is primed and the clonal expansion is produced. But when CTL4 is upregulated in the lymph node, it blocks the clonal expansion. So once the T cell is activated, it goes to the tumor again to find the antigen and to kill the tumor cells. So the engagement of PD-1, PD-L1 attenuates the response. And PD-1 is expressing T cells, and PD-L1 is expressing the tumor in the microenvironment. So CTLA-4 and PD-1 and PD-L1 play different roles during immune response, and they are known as immune checkpoints. And we are going to see why it's important to block them. So as you may imagine, up to here, it's important to have tumor antigens because they activate the immune response. But how are tumor antigens generated? Tumor cells accumulate changes or mutations due to their high rate of proliferation and their inability to repair DNA mistake. But the important thing is, even when they only few changes are drivers event, that means that can begin a new tumor, the accumulation of this mutation may enhance the immunogenicity and of course may change the immune landscape of the tumor. So neoantigens are very important because they awake the immune response. They are generated by mutation, and we now can define the tumor mutational burden, that is the number of mutation in a tumor. So you can have low or high TMB tumors. And very importantly, the more TMB, the more neoantigens, and the better recognition by dendritic cells. Mutations in genes coding proteins, mismatch repair proteins, increase the TMB. So because if you cannot repair them, you have many errors and you have many mutations. And tumors with high TMB, high MR mutation, and high microsatellite instability are candidates for immunotherapy. But as you may know, and it's very known about immunotherapy, that not all tumors will respond to immunotherapy regimens. But we can predict which tumor will respond, and we know that the number of TILs and as other infiltrated immune cells are important to respond, that the PD-L1 expression in the tumor is very important, and the defect in the DNA mismatch repair, the high TMB, the macrosatellite instability, always promote the response to immunotherapy. But one question is very important, and is how many mutations I require by a tumor to respond to immunotherapy. And there's a critical cutoff of mutation, and that number is near 192 mutation per tumor. This strange number came from this study where they compare the number of mutation in 26 types of tumor, and they compare the response of those tumors with the melanoma tumors and the lung tumors that are the better responders because they have many mutations. And for example, here within the lung tumor group, you have the tumors from smoker persons that has more mutation and responds better. And here we have the breast tumors that are exactly in the middle, with only 15% of the patients harboring more than 50% harboring more than 182 mutation. And this is why the immunotherapy doesn't work a lot in breast cancer, okay? So breast cancer is not very sensitive to immunotherapy with immune checkpoint inhibitors due to its low immunogenicity. Within breast cancer, we have 3 big groups that are very different. We can say that we have luminal tumors that has hormone receptor expression as progesterone and estrogen receptor. We have another group that is HER2 positive that overexpress the oncogene ERB2, and we have a third group that is a basal or triple negative group that do not express hormone receptor and do not express does not express the oncogene HER2. And these both HER2 group and the basal or triple negative group are the most aggressive tumors. So apparently, the HER2 group is the group with more mutations, with that has a more TMB, but is low also. As you can see here, it's only 13%. And why there is a difference among subtypes in breast cancer? Because, as I told you, each subtype is different. It has different markers, it has a different overall survival, it has different treatment, and of course, it has a different response to immunotherapy. Of course, always TILs are markers of good prognosis in all groups, but are more frequent in triple negative and in HER2 positive groups. Luminal tumors that are hormone responsive tumors have low TMB and low cytotoxic infiltration, but have higher Treg frequency. And triple negative groups has the highest expression of PD-L1 among all cell types. And that is why it may also respond better to immunotherapy. This is the first evidence of that. About 19% of the triple negative breast cancer patients express PD-L1, and the expression of PD-L1 was associated with the loss of the phosphatase P10. And this was associated also to CD8 infiltration. So this led to the approval of immunotherapy for this group of tumor, for the triple negative tumor 2 years ago. And the FDA approved the application of atezolizumab in combination with nab-paclitaxel for advanced triple negative breast cancer, the improvement on the response was modest, but of course there was nothing better. So the enthusiasm was big, but the response was low. And that means that all we need is work harder on that to see how we can improve the immunotherapy response in breast cancer. Some months ago, the FDA also approved the combination of pembrolizumab with chemotherapy to treat triple negative breast cancer. So a good way or a good strategy to make a tumor respond to immunotherapy is to turn a cold tumor in a hot tumor. Of course, breast tumor is not a desert tumor. It has infiltration, but we need to infiltrate more, to have more cells in the tumor bed. So because as I told you before, breast tumor has they lack of antigens, they have absence of T-cell activation, they have the impaired trafficking of cells. So if you can reverse this situation and you can turn a cold tumor in a hot tumor, maybe you can have a better response. So you can do that by combining immunotherapy with chemotherapy, with radiotherapy, with targeted therapies as inhibitors of the CDK4 and CDK6, and you can deplete, for example, suppressive populations as regulatory T cells, macrophages, or myeloid-derived suppressor cells. And the talk in this talk, I will talk about the possibility of combining immunotherapy with endocrine therapies to see if we can improve the response to immunotherapy. Okay, our first aim was not to see if the combination works, because we originally wanted to study the contribution of the immune system to the anti-progestin-induced tumor growth inhibition. We have a tumor that was sensitive to the treatment with anti-progestin, that is an inhibitor of the progesterone receptor. And when you treat this tumor with mifepristone, that is an anti-progestin, the tumor regress. So we wanted to see if this happened equally in an immunocompetent or an immunosuppressed host. So we generate chimeric mice using not-skid mice that were injected with green bone marrow of a bulb C, that is GFP bulb C. We took the bone marrow from those mice and injected in the not-skid, and we generate fully immunocompetent mice in 2 months, more or less, that have a green immune system. So we evaluate if those mice were full immunocompetent, and we can confirm that. We detect in the bone marrow and in the blood green cells, and there were also CD4 and CD8 cells in the blood, and we can detect CD8 central memory as CD4 effector memory T cells in circulation in those reconstituted mice that were comparable or were similar to the parental green mice or the wild-type bulb C. Cytokine production was also okay and was comparable between the parental mice, and the reconstituted mice recovered spleen size. But more importantly, they recovered the ability to reject a xenogenic pancreatic tumor. So they were immunocompetent. So we compare if in those mice that were immunocompetent or in not-skid mice that were immunosuppressed, the mifepristone was effective to inhibit tumor growth. We also use a second mouse model that was the wild type BAL-C, treated or not with mifepristone. So we have this that was immunocompetent, this that was immunocompetent but green, and this that was immunosuppressed. So as you can see here, you first pay attention to this graph. Mifepristone was able to inhibit tumor growth in the BAL-C mice very effectively. But here, when we compare the presence or the absence of the immune system, mifepristone was always effective to inhibit the tumor growth. So we were a bit disappointed because we thought that maybe in the presence of an immune response, mifepristone would work better or worse, but anything but nothing happened. It was the same. But then when we check the tumors, we were happy again because we have an effect. When we treated with mifepristone, the tumor was different. It promotes changes in the tumor immune landscape. It promotes remodeling that was associated with an infiltration of green cells, of GFP positive stromal cells. And that cells were CD45 positive, and they were CD8 T cells. Mifepristone also induced the infiltration of NK cells. So something happened when we treated with the antiprogestin the tumor. Importantly, mifepristone also promotes a reduction in suppressive populations. As you can see here, it reduced the frequency of regulatory T cells and the ratio of regulatory T cells CD8 T cells. And those regulatory T cells have less expression of CTLA-4. And as we can see here, the myeloid population was reduced. The myeloid-derived suppressor cell population was reduced when the tumor were treated, both in the tumor and in the spleen. So it was very important because suppressive population were reduced locally and systemically. Well, moreover, mifepristone also induced an increment in the proportion of cross-presenting dendritic cells that were CD103 positive. You see here, we can see the increment in this population. It also induced an increment in the lymphocytes that were PD-1 positive, and an induction in the filtration of macrophages that were MHC2 positive, and were CD206 that were like M2 macrophages. But more importantly, they were PD-L1 positive. And remember that we are going to see this part at the end of the talk. So up to here, we have that mifepristone inhibits tumor growth, but also during inhibition, it promotes infiltration of T cells and it promotes tissue remodeling, and apparently an activation of the immune response. So we wanted to see which transcriptional programs are activated by mifepristone. For that, we did RNA-seq in treated tumors and untreated tumors, and we observed that there were many genes that were up-down regulated with mifepristone. But if we analyze those genes, we determine that mifepristone upregulate biological processes associated with inflammation and immune pathways. That is very important for the effect we are seeing. But what was really good was to find that mifepristone regulated a pathway that was associated with immunogenic cell death. That is, it was like a good point to activate an immune response, immunogenic cell death, instead of other type of death. So as you may know, immunogenic cell death favors antigen presentation in lymph nodes and is mainly mediated by the exposure of calreticulins and other proteins that are exposed when the cell is dying. And this situation, this exposure, activate the maturation of dendritic cells. So those dendritic cells now better present antigens and activate the immune response. So we evaluate if this was happening here. And as you can see here, all those tumors that are sensitive to mifepristone when they are treated in vivo with mifepristone, they upregulate or they expose calreticulin. So if this has an effect, we evaluate it here when we co-culture those cells with immature dendritic cells, and we evaluate the maturation stage. And only dendritic cells that were co-cultured with tumors that were sensitive to mifepristone were able to activate dendritic cells. We also evaluate if the endocrine therapy may activate T-cell lymphocyte in an antigen-dependent manner. For that, we took tumor-bearing mice, treated or not with mifepristone, and we purified T cells and we confronted T cells with the tumor antigens, and we evaluate the activation state of those cells by measuring the interferon gamma production and the CD69 expression. So as you can see here, interferon gamma production was induced in CD4 cells, and in CD8 T cells, we were able to observe an activation state. So we have up to here an activation of the immune response that is apparently mediated by an immunogenic cell death that remodulate and that promote an infiltration of T cells. So we wanted to see if this situation may protect the mice against a subsequent tumor challenge, that in the case of breast cancer would be the same to have a metastasis, because when you have the primary tumor, you are operated, and sometime later you can have a metastasis. So it's important that this is like a secondary tumor challenge. So mice were treated or not with mifepristone, and then tumor were removed, and a second tumor was inoculated. And only mice that had been previously treated with mifepristone were able to reject a subsequent tumor challenge. And this was specific because if we change the second tumor to a colon cancer, colorectal cancer cell line, it was the same. So mifepristone induced an immunological memory. And finally, I want again to show you that mifepristone also induced T-cell infiltration, but also induced the expression of PD-1 and PD-L1 in the stroma. Remember, I show you here that CD4 T cells that infiltrate the tumor were PD-1 positive, and that macrophages that were in the tumor bed with PD-L1 positive. So what we had is a therapeutic opportunity, because we thought, okay, now we have the conditions that would predict the response to immunotherapy. We have infiltration, we have CD8 cells, and we have PD-L1 expression. So we at this moment we said, okay, let's combine the mifepristone with the immunotherapy with anti-PD-L1. We chose the atezolizumab because it was the one that was approved for triple-negative breast cancer. And as you can see here, the treatment with anti-PD-L1 alone slightly inhibit tumor growth, but was not significant. But when it was combined with mifepristone here in the combo, the effect was really hard. We use here a low dose of mifepristone because we didn't want to completely inhibit tumor growth to see an effect in the combination, no? And as you can see here, the effect was immune mediated, and you have here an induction in the CD8 Tregs radio. You have a reduction in Tregs, an induction in memory CD8 T cells, and an induction in the production of cytokines by CD8 T cells, and some of them here with in CD4 T cells. So, well, the idea was that to mix two topics, to mix endocrine therapy with immunotherapy, and to show finally that antiprogestin therapy induced immunogenic tumor cell death in tumors that overexpress progesterone receptor A. This elicits an activation of the immune memory response that protects mice from future tumor recurrency and increases sensitivity to PD-L1 blockade. So you can see here that we turn a cold tumor in a hot tumor, and that this in combination with immunotherapy make a good response in luminal tumors that are that up to now are out of the immunotherapy options. So the last slide is only to highlight some points that I want you to remember, is that tumor with low or intermediate levels of immune infiltrate may take advantage of previous treatment with endocrine therapy. That hormones may regulate the anti-tumoral immune response in tumors that are hormone receptor positive or hormone receptor negative, since many immune cells express hormone receptor. This is very important because when you have a patient that have a triple negative tumor, they say, okay, don't worry, it's not important if it's a menopause or not, because the tumor is not an hormone receptor positive, but immune system does respond to hormones, and this is very important. And finally, the same message I told you, antiprogestin therapy induce immunogenic cell death that activate the immune response and protect the mice from recurrency or increase sensitivity to PD-L1 blockade. And this is the paper if you want to take a look. And this is, well, Gabi, I think you know him, that was the director of the lab. And this work was in collaboration with another lab in the IBYME, that is Dr. Lanari lab, and well, Tomás Dalotto, that is my first PhD student. Now he's a researcher, but he was the first author, and Ramiro, that is also my PhD student. He did all the bioinformatics study. So this was the idea of the work, and thank you for the attention.
Thank you so much, Mariana, for an excellent and very exciting presentation with really, really interesting possibilities for treating breast cancer. We do have time for one question, and I'd like to pass it on to Ken, because he already sent me a message here saying that he'd have a question for you. So please, Ken.
Thanks a lot, Mariana, for the nice work. So I have a question about, I'm not sure if in a model that you use, you're able to look at the question of time, of how long. So if you think of somebody with let's say a tumor microenvironment that does not have a lot of infiltrating cells, and you give your hormone treatment, the the MFP treatment, I could imagine that there'll be initial activation that may precede the induction of PD-L1 and PD-1, CTLA-4, etc. And my question is if we have a good idea of what timing would be like between the induction of the activated immune response and the antigen presentation, and starting with your anti-PD-1 or anti-PD-L1 therapy.
Okay, thank you. Thank you for the here. No, I can't turn on my camera. I don't know. Yeah, initiate my video.
We can hear you though.
Yes.
Okay.
Yes. Yes, great question. We don't know exactly. We treated tumors for 1 week, and then we observed the effect after that. So maybe it's a rapid effect. The modification of because mifepristone induced a radical regression, like it's immediately you treat the mice that these tumors are very sensitive to mifepristone. Because of that, you can modulate the inhibition so easily. And so when you treat the tumor with the mifepristone, you have the effect right in that moment. The tumor regress, and in the remaining tumor, you have those infiltrating. You have the increased infiltration, and you have in those in the microenvironment, you have the induction of the PD-L1 expression, and the but this is very quick. No, it's within a week.
Yeah,
I don't know if
because
in humans would be the same, of course.
Yeah, just because we usually will expect to see high PD-L1 in endothelial cells and tumor cells when you have the high gamma, high Th1 type response happening. And it's interesting that it's being induced, but shortly after that, I imagine it's being suppressed. And so I was thinking about timing as to, well, I mean, we'll have to see what happens in trials in humans, of course, but it's a very interesting dynamic. Yeah, very nice.
There is a trial
ongoing,
but only with mifepristone that patients that have luminal tumors with high ratio of progesterone receptor A can go to mifepristone treatment, and they are treated for 15 days before the operation, and they are operated. And they do not evaluate any immune point or any immune outcome, because the trial was planned to see only security and mifepristone effect on tumors. So maybe in the future we are very interested in designing a new one.
Okay, Mariana, thank you so much for excellent presentation once again, and for being here with us. And I will now turn it into Gisela Suárez, Dr. Gisela Suárez, who will introduce Ken for the next presentation. Thank you very much, Gisela, for accepting our invite to be here with us today representing Cuba. Thank you.
Okay, thank you, all of you. It's a pleasure to be here with you at this time. But to continue, I present to you an excellent professor in science, Ken Gollob, who attended his PhD from the University of Colorado Medical Center, performed his postdoctoral training with Robert Kaufman at the Nax Research Institute. Currently, he's a senior scientist and head of the Transnational Immunology Laboratory at Hospital Israelita Albert Einstein in Sao Paulo, Brazil. So his research focuses on using multi-omics approach to elucidate immune mechanism behind disease progression and response to therapy in cancer and infectious disease. He has over 100 publications and has trained over 30 graduate students and postdoctoral fellows. So it's an honor to share and learn from him today. Professor, thanks for this opportunity, and the word is yours.
You're mute, Ken.
Yes.
Yes, thank you. Let me thank you again, Gisela. Thanks for the nice presentation and the invitation from SBE, from Val, from IMMUNOLAC. It's nice to be able to share our work with you all today. Let me share my screen. Okay. All right, we're seeing the screen well.
Yes.
Okay, so today I'm going to share with you some work we've been doing on trying to identify immune markers that can indicate therapeutic response or development of adverse events in patients treated with checkpoint inhibitors. And I'll go into some detail as to why that's an important issue to address today. So very briefly, just as a reminder, the immune response can play a role in the formation of cancer in the form of inflammatory background environments that can lead to a higher chance of developing cancerous cells. And of course, there are many other important factors such as genetic susceptibility, DNA alterations due to mutations, environmental factors, and infections with viruses that can lead to cancer formation. And where I focus my research is on this point here, where you have individuals who have already developed a cancer. And in the best case scenario, of course, that cancerous cell will be detected by the immune response, eliminated. This is related, as Mariana went over very nicely, to hot tumors and tumor mutation burden and genetic instability. And in this case, you have destruction of cancer cells, and the person never even knows they had a cancerous cell. However, for individuals that arrive at Einstein or Sacramento Cancer Center, where these studies I'm going to show you were done, in those cases, the immune response was ineffective at recognition and elimination of the cancer cells. And this can be due to many different reasons, ranging from inactivated T cells to suppressor cells, suppressor myeloid and microenvironments because of fibroblast cells as well, and many other mechanisms. When you have a patient that has a tumor that is suitable for treatment with checkpoint inhibitors, we're able to reactivate the immune response to combat the tumor. And this has led to increased survival in many individuals. So again, I'll go over this very briefly since Mariana did a nice job already of talking about this. We have different tumor types ranging from cold tumors, which have very little infiltrating cells, and they can either have fibrotic nests and barriers to access, or can be due to really low neoantigen load as well, or a combination of these things. Then you have hot tumors, which are the two I'll be focusing on today, melanoma and lung cancer, which are excellent candidates for treatment with immune checkpoint inhibitors to reactivate immune response against neoantigens. So as a brief reminder in the context of melanoma and metastatic non-small lung cancer, I'll briefly remind you about the activity of CTLA-4 and PD-1. So CTLA-4 is induced upon activation of T cells. Of course, we have signal 1 and 2 leading to the activation of a cell. Shortly after activation or due to chronic activation, you get expression of CTLA-4, which then sends a negative signal, reducing proliferation and activity of this cell population. So this is the work of Jim Allison in the 90s that led to immunotherapy with anti-CTLA-4. And of course, the Nobel Prize in 2018 was awarded to Allison and Honjo. And Honjo's work was in the area of PD-1 and anti-PD-1 and PD-L1 therapies, where again, you have activation of a T cell, induction of PD-1, which leads to a negative signal, turning off cells that could potentially combat the tumor cell. And their ligands are PD-L1 and PD-L2, which can be expressed by many different cell types, but in particular in response to an inflammatory microenvironment. So these are natural, important parts of immunoregulation that are important for reducing autoimmune diseases and autoreaction and dominance of one given T cell population over the other T cell populations in the body. However, in cancer, they become a problem because they can inhibit an anti-tumor response. And here's just a demonstration of the success of immunotherapy in melanoma, where the 5-year overall survival with chemotherapy was around 10 to 20% before immunotherapy. And today we see survivals 52% or higher at 5 years using a combination of anti-PD-1 and anti-CTLA-4. For metastatic non-small cell lung cancer, also a devastating disease, we see the 5-year overall survival with chemotherapy was only 5.5%. That was raised to 23 to 50% with anti-PD-1. And today, the patients I'll be showing you in my cohort were treated with anti-PD-1 and chemotherapy, where we see a 12-month survival of around 70%. So these have brought great benefit to the patients. However, there are still many challenges around checkpoint inhibitors. And the main one is that not all cancer patients respond to immunotherapy checkpoint inhibitors, and some of them also develop very severe autoimmunity. So this is both important to not be treating a patient with a therapy that won't necessarily work well for them, but also to try and make checkpoint immunotherapies more available to the general population. In Brazil, the public healthcare system, SUS, does not cover the use of checkpoint inhibitors in patients, even when it's an indicated treatment. And so that, in part, is because of the high cost of these therapies. So if we can target these therapies to patients who will most benefit through the identification of markers that can identify those patients, you could bring costs down dramatically. Our studies, these studies I'm going to be showing you today, were carried out at São Marco Cancer Center. I recently moved to Hospital Israelita Albert Einstein, where we're continuing these studies and expanding others. And they're really available because we have quality clinical data and expertise in patient care. We have the scientific know-how in the laboratory using some multi-omics approaches for studying the immune response. And of course, the patient volunteers who agree to participate in the donation of a blood sample and then a fragment from tissue when available. And again, our objective is to discover immune mechanisms behind therapy failure and also biomarkers for novel identification of these patients that will respond or not. So taking all this information into the context of immune response, we have a couple of different directions we can go. When we have an effective immune response, we can, in the best case, lead to elimination of the tumor. And this is characterized by this activated inflammatory response that can be identified by many different markers, but in particular, I'm highlighting here interferon gamma, CD8 positive tumor infiltrating lymphocytes, CD4 Th1, M1 macrophages, for example. On the other polar end, we see a suppressive immune response that's characterized by exhausted T cells. Many different markers can identify those cell populations. Regulatory T cells, Treg cells, IL-10, a suppressive cytokine, M2 macrophages. And so the checkpoint therapies then act to try and take this response and drive it back over this direction into an activated response. And Mariana mentioned many interesting approaches to try and activate an immune response to drive it towards an inflammatory response. There's been studies done with TLR agonists, like SD101, that activate TLR9 to try and activate microenvironments in head and neck cancer. And that sort of treatment together with checkpoint inhibitors may be an important way to increase the response to therapy. But given today that we have the majority of patients treated with checkpoint inhibitors alone or checkpoint inhibitors plus chemo or a targeted therapy, we want to try and identify markers that can really identify those individuals that will respond and those that will not develop these severe adverse events. So what happens here is obviously we're reactivating T cells to combat a cancer, but when we do that, we can also reactivate autoreactive T cells, which leads to an adverse event in the form of autoimmune diseases. Those can range from mild autoimmunity to severe disease that requires suspension of treatment. So just as a brief reminder, lung cancer is a major problem, as well as melanoma in Brazil and globally, with around 8.7% of cases of cancer in men and 6% in women. And melanoma is 12th position for men and 13th for women. So this is our study design. We are still enrolling patients in this study, but we've reached an N of 40 in this group. And this group, we're continuing beyond the 40 because of the high response rate. So we actually had a low number of patients in the non-responder group, which was great news. These are patients with non-small cell lung cancer that have PD-L1 and greater than 50% of the cells and treated with anti-PD-1 and chemotherapy, and melanoma, the combination therapy of anti-PD-1 and anti-CTLA-4 for the great majority. So what we did at time 0 was collected blood as well as a tumor biopsy when available. And I'm going to focus on the blood work, looking for blood biomarkers. Again, this is a more useful type of biomarker when you're thinking of clinical utility. And so we focused our studies for biomarkers in the blood. And I'll be showing you information from time 0 blood draws. So this is blood drawn before the patient has started their immunotherapy treatment. And the idea, again, is to look and see if there's some indicator in the blood, an immune indicator, that will predict which patients will respond or which won't, and could also help shed some light on possible immune mechanisms behind failure or success of treatments. We'll be looking at patients that have fallen into responder or non-responder groups in what we call a rapid responder for the non-small cell lung cancer studies, or durable responder, again, for the non-small cell lung cancer studies. So for the responder, non-responder work, I'll be showing you just our data from non-small cell lung cancer. And then from for our adverse events, I'll show you a combination of both cancer types. And we looked at a series of markers looking at T cell populations, activation, exhaustion, as well as monocyte subpopulations, and again, other markers for activation or exhaustion. So to do these studies, we use high parameter flow cytometry. As a brief reminder, flow cytometry allows us to mark cells, to stain cells with fluorescent markers. These can be for surface markers and all sorts of functional activity. These cells are then run in flow in front of a laser or lasers. In our case, we have 5 lasers and 28 detectors using the FACSymphony A5 machine. And so this really increases our power to be able to look at many different cell populations in a very small sample size. Of course, the flow cytometer was invented by Lynn Herzenberg, and in 2006, when we were actually in his laboratory as a visiting professor, he won the Kyoto Prize for Medical Technology, which was a real well-deserved achievement for the Herzenberg group. So upon analyzing these different colors and markers, we can analyze those manually or using computational methods. So very briefly, the power of the high-dimensional flow cytometer is, as I mentioned, lets you look at many different markers simultaneously. And we perform a series of standardization steps called filtration, where we use anti-CD4 stained with all of the different possible fluorochromes, and we look at their signal across every single detector. So here we're looking at the BUV661 fluorochrome and its detection or not in all of the different detectors across all the different lasers. And this lets you identify combinations that you want to avoid. So anywhere where you see spillover, you'll see this kind of spread out pattern of light entering that detector. So here we see BUV661 entering the detector of APC. So this is a combination we would want to avoid with molecules that are expressed on the same cell or expressed at high levels. So all of this leads, this was done by Clara Cavalcanti, who's our excellent operator of the FACSymphony, and Amanda Braga, postdoc, was with me at Second Margo and now has joined me at Albert Einstein as a visiting scientist. This lets us generate this, which is called the matrix spread. And with this diagram, we have all the different dyes and all the different detectors across the top. And it allows us to see what combinations are very safe to combine with markers that are expressed by the same cell type or in high concentrations, and those that you want to avoid where you'll have bigger problems with compensation. Of course, you can always use compensation to try and reduce spillover, but what we try to do is design the panels to just take advantage of all these white areas in the compensation matrix. So we perform these studies and using these panels, and analyzed two different groups. So here what we see are non-responder non-small cell lung cancer patients and responder non-small cell lung cancer patients. Again, these are 10 patients that were in the non-responder group and 15 in the responder group. We joined together both stable disease and partial response for the what we're calling the responder group. And this analysis is done using TISNI, which is a machine learning non-directed methodology. So these populations that are identified using TISNI were not chosen by the researcher. We can do both our own directed analysis, where we choose a cell population and we calculate the frequency of that population. But what's done here is the program itself forms clusters in both the non-responder and responder groups, and then we look and see what those populations are. And so what I've highlighted for you here, just as an example, is this population that's present in the responder here in green and missing in the non-responder. And you go and look and see what that population is. It was activated CD4 positive T cells. So we saw an increase in activated CD4 positive T cell cluster in the responder compared to the non-responder at the level of the TISNI analysis. So we go on to then perform a statistical analysis of these data and see which findings are statistically significant and which aren't. And you can see here two that I've highlighted. Here on the left-hand side, we're looking at a population that was present in the non-responder and less present in the responder. But you can see that this is a non-significant difference between the non-responders and responders. And this was total CD4 positive T cells in the blood, again, before the patient was treated. So we're looking at response now 9 weeks after treatment. And then the activated CD4 positive T cells that I showed you in that earlier slide, those gave a significant difference, being much higher in the responder compared to the non-responder. So this says that peripheral blood activated CD4 T cells seem to be increased in patients that are going to go on to respond at 9 weeks. We ran some area under the curve analysis, and combination of the CD4 and the CD4 CD69 positive activated T cells led to an area under the curve of 0.85 and a strong p-value in the ability of these two markers to predict which patients are going to respond at 9 weeks to the anti-PD-1 plus chemotherapy in the non-small cell lung cancer group. So we went on to look at many other populations. One of the others I want to call attention to in this talk is the one that we found in monocyte populations. So monocytes can be divided into classical, transitional, and non-classical monocytes. They have functional differences, but again, here we're using them as a marker. And we can see that the responder group had a much higher frequency of the non-classical monocytes in the blood compared to the non-responders. And this gave a quite high area under the curve analysis as well for predictability of responders versus non-responders. Again, this is at 9 weeks. So we went on to look at 6 months. All these patients in this study that are shown here have all had at least 6 months of therapy. And we found that some of the markers that we found in 9 weeks were maintained and others weren't. And in particular, this combination of the percentage of CD4 positive T cells and activated CD4 positive T cells was the best combination for predicting response at 6 months after therapy began. So the non-classical monocytes fell in their significance and were no longer a strong predicting population at 6 months. So this really tells us that the ability to look at markers early on before the patient starts therapy may serve as an important tool for being able to identify which individuals will go on to respond and which won't. And this is looking at survival curves of the same groups of patients based on now the expression of those markers I just discussed with you. And I'd like to just focus down here on these bottom two figures, which is the combination analysis of total CD4 and low levels of the activated CD4 had the worst survival compared to all the other combinations. And those individuals that had high CD4 and low of those monocyte populations, remember the monocyte, non-classical monocytes also had the worst survival compared to all the others. So as a potential marker that we of course need to validate in a much larger study, But in theory, the idea here is that you could use these markers to identify people that maybe aren't such good candidates for this therapy, or ones that need some other combination therapy in order to have a better survival. So in summary of these results, then looking at potential markers for patients with non-small cell lung cancer treated with the anti-PD-1 and chemotherapy, we see that those that go on to respond with a durable response have an increase of activated CD4 positive T cells compared to the non-responders, and they have lower resting CD4 cells. And for early response and also for combinatorial analysis, the non-classical monocytes are also a good indicator of those that will respond and have better survival. For those that have therapeutic failure, it's the opposite, of course, with a higher frequency of resting cells, lower activated CD4 positive T cells, and low numbers of non-classical monocytes. These actually fit well with what we think of in terms of some of the mechanisms of immune activation induced by anti-PD-1 and chemotherapy. And we have many other studies underway looking at detailed functional activity and the exhaustive state of many different cell populations in the same patient group. But this basically told us then that this durable response is predictable by an activated circulating immune response in terms of CD4 positive T cells and non-classical monocytes. So I'd like to finish with a couple slides showing you the design of these adverse event studies and show you some preliminary data that we've gathered from this group. So again, as I mentioned, one of the side effects of checkpoint inhibitor therapy that can lead to suspending therapy and in the worst cases, of course, can lead to life-threatening autoimmune diseases, are the activation of cells that can have autoimmune reactivity. And with the different immunotherapies, we have different frequencies of induction of autoimmune activities, with anti-PD-1 being the least, the lowest frequency of adverse events, anti-CTLA-4 next, and combination therapy having the highest frequency of adverse events. And we want to try and identify as well, immune components can predict which patients will be in the group that either has very high adverse events or very low adverse events. So in this study, we're talking about time 0, and then we're analyzing adverse events within 6 months after therapy. Over 85% of the cases of adverse events happen in this window. And again, we have peripheral blood analyzed using flow cytometry as well as systemic cytokines, but I'll be showing you the flow cytometry data today. So this is a combinatorial analysis. One of the things we wanted to do is see if we could identify a cancer type agnostic indicator of individuals that develop severe adverse events. And we have in this study around 19% of the patients that develop severe adverse events, and 64% with G0, which is no adverse events, and 17 with mild adverse events. So these don't really affect the progression of the therapy. And this work was done by Guilherme, who's a master's student in the lab. The previous work was by Amanda, a postdoc and currently visiting scientist at Einstein. And what we saw here was that the patients as a group, as a whole, with the no mild versus severe, had a higher frequency of total CD4 positive T cells and the no and mild adverse events compared to the severe. And those that had severe disease had a higher frequency of total CD8 positive T cells. And then when we looked at the area under the curve for this combination, we saw that CD4 cells did a little better than the combined CD4 plus CD8 in this combined group. Now, remember, we're continuing these studies with a much larger N to increase the number of severe and both melanoma as well as non-small cell lung cancer patients. So this is preliminary data, but it shows you an idea of what we're doing in these studies. So in summary, what I wanted to show you today was that this question of not all patients responding to immunotherapy, we may be able to identify that via blood tests in non-small cell lung cancer treated with anti-PD-1 plus chemotherapy, as indicated by an increase of activated peripheral blood CD4 positive T cells in the responder group and non-activated CD4 T cells in the non-responder group. And those individuals that go on to develop autoimmune disease may be characterized by this higher frequency of CD8 T cells in the blood and a lower peripheral CD4 positive T cells. So we, of course, are expanding this study. We have a lot of other analysis underway currently. And this data, we're continuing to look at other cell populations, including exhaustion cell markers, activation, and functional activity markers in different cell populations. So I would like to acknowledge the collaborators in all these studies from the Second Margot Cancer Center, where I spent 4 years, established the immuno-oncology group, and together with an excellent group of clinicians that are highlighted here in non-small cell lung cancer and melanoma, as well as diagnostics and radiology, surgeons and oncologists, the immuno-oncology group at the Second Margot at the time, and Clara is the person who was responsible for the FACT Symphony flow cytometry operation, as well as graduate students and nurses that worked on the study, and the group that's now also with me at Albert Einstein. All these individuals were over at a Second Margot as well, or still at a Second Margot, working in both locations. So I also have collaborations with Walderez Dutra at UFMG, Georgia Trinquete at the NCI, and Andrea at Fiocruz, and our funding agencies from FAPESP, NCI, CMPK, and CAPS. Thank you for your time, and I'm happy to address any questions or comments.
Well, thank you, Ken, for that excellent conference. We think that it's very important to find markers that help us to identify those patients who will have a better response to anti-tumor therapy. So we have time for one questions from Wal, please, can you ask your questions?
Yes,
I don't know where it's, okay.
Yes, very, very interesting work, Ken. It's every time that I see you talking about it, it's a new opportunity to learn something. And one thing that I was curious is that I do understand your approach of looking at frequencies of activated CD4s and macrophages and CD8s as markers of to try and predict who would be a good or a bad responder to the immunotherapy. But my question, I have two questions actually. One is, what do you think is causing the low frequency of activated CD4 in these people that will be no good responders? And the other question is whether you have any indications as to the functional aspect of these CD4 cells that are in lower frequency in the no responders. Would they be the ones that would be responsible for activating the inflammatory profile, or do you have any data on the functional aspects of them?
Yeah. So yeah, thanks, Val. And these are things that we either have underway in studies that are ongoing. But in terms of the general concept, I kind of think about it a little more in the direction of those patients that respond have this higher frequency of activated cells. We of course need to know what their functional potential is via cytokines and other exhaustion markers and co-stimulatory markers. We just performed a series of single-cell RNA-seq studies on these patients, which will, together with the T-cell receptor CDR3 region. So those studies will be key for identifying either a clonal expansion that's present in the blood. And we also have in those studies metastatic lymph node from the patients from the same patient. So we have blood and lymph node with the T-cell receptor CDR3 and functional potential. So that will tell us a lot more to address your question. Of what our snapshot in the blood, you can imagine that you have cells that were activated in a node, and they've fell into the circulation and are transiting to a tissue, right? They're on their way to the lesion tissue. So we have a snapshot there that could either be super underrepresented compared to what's going to be in the tumor infiltrate, or it can represent well what's going to be in the tumor infiltrate. Our studies in human leishmaniasis, as you know, have been extremely reflective of what happens in the tissue. So what we see in the blood, we see in the tissue. There are other studies that we've carried out where we see differences between the blood and the tissue. So really, we'll be able to identify those factors soon. And in terms of what those cells are doing, we have to wait for the study. I anticipate since they express CD69, we'll also see other activation markers and cytokine profiles, but we have to see which cytokines those are. We have some other panels now that we've run with around a total of 40 different markers that will really dissect all these questions of T cell exhaustion, functional potential, and cytolytic activity. And then we'll know more about that. But thanks for the question.
Okay, Ken, we have another question for the audience. Carolina wants to know if you evaluated exosomes expressing PD-L1 in these patients.
Yeah, that's a great question. There is some really nice work, especially in melanoma, studies that I know of. I'm sure there are others of the importance of exosomes to be able to either, many different activities, either condition environments for metastasis, or even alter the immune response in condition microenvironments. So we haven't looked at that. We have plasma frozen down from all these patients at -80, and we've been speaking with some collaborators that potentially could look at that with us. One other set of studies we've been doing is metabolomics in collaboration with Fleury, and they have a good metabolomics group that's been looking at many different metabolites that may shed some light on other interesting markers. So far we haven't done anything with exosomes, but that's a good point.
Okay, Ken, thank you very much. And maybe the last question is about if you know activated CD40 cells produce also granzyme and perforin in these cases.
Yeah, so I'm very excited for us to finish the analysis of these new panels. I didn't have, because of time constraints, I have panels that will be able to look at all that for us, both granzyme expression, cytokine expression in CD4, CD8, double negative T cells, and we're seeing some pretty consistent correlations between CD107A, which is an indicator of degranulating lymphocytes, as well as granzyme. We have both markers. So I think there will be, again, I'm talking about the non-small cell lung cancer. It looks like there'll be a pretty strong correlation for those activated cells. We haven't finished all the analysis yet, though.
Okay, that's really great. Thank you very much, Ken. Okay, well.
Okay, so I just would like to finish by really thanking Mariana and Ken for the brilliant presentations. It was truly excellent opportunity to be here. And even for us who work with parasites and metastasis in the diseases that we work with, and dormancy and overriding the immune response, it's always very interesting to see your approach, and it really enlightens a lot of the research that we do in the different areas. I think that really is a great thing in research, is to be able to hear people and their insights into something that is different than what we do. So thank you both very much. And my special thanks to Gisela for sharing the moderation with me. Thanks a lot for coming. And also thanks to Rafaela for giving us the support, Rafaela from SBI for giving us the support for this talk and for the announcements and all that. And of course, to all of you present in the audience, I'd like to particularly thank the presence of Emilio Malchiodi from the Argentinian Society of Immunology, and the current president of ALACI, as well as Professor Manuel Barral Neto, who really is the idealizer of IMMUNOLAC and has been in very important support for this presentation. I hope to see everyone back here in December. We are going to have the 3rd edition, and the subject will be mucosal immunology. So I think it's going to be quite exciting. So thanks, everyone, and I'll see you in December.
Thank you.
Okay, thank you very much.
Nice to meet you.
The same, nice to meet you, Ken, Gisela, and Walt. Thank you very much.
Thank you.